Neuroplasticity can make subtle little local changes in a brain, but it can't just change the entire brain, okay? It's like improving traffic flow in a city, okay? Putting in some yield lanes, yield turns or some traffic circles or something. That's neuroplasticity, all right? It's not leveling the entire city and redesigning the whole thing from the ground up. That is not neuroplasticity. That's not how it works. Hello and welcome to the Trauma-Informed SLP. I am Kim Neely. I'm a licensed speech-language pathologist. That's what the SLP means. And also neurodivergent and queer. And welcome. Here we are. We're going to unpack stuff today. That's what I'm hoping for. So I saw this show up on one of the subreddits I follow. It might have been Our Psychology or maybe Neuroscience. I'm not sure. I think it was one of those subreddits where people typically present research that they want to talk about. And then I did also see it somewhat show up on Our Autism, I think it looked like somebody from TikTok who was claiming that like this research group essentially stole her work, like didn't cite her, basically based the study on her work and then didn't cite her, which is a really big faux pas in academics. That is a really big deal because it's essentially effectively, especially if you're taking it from a like graduate student or someone who hasn't started a career as a researcher, you're effectively suppressing their career at that point because citations are freaking everything when you're first starting out. And so like being cited by a big study with like, this is a Stanford study, so big lab, big study would have meant a lot. And it's really BS not to cite. And it's also plagiarism. That's worse than academic shade, okay? That is bad. But I did see this come out and I wanted to unpack this a little bit for people out here because this is exactly the type of article that I had to read for my neuroscience classes when I did the PhD level classes. And when you're a graduate student, a lot of the article readings you do for any sort of class, they're sort of this vibe of article. And the whole goal of the reading, you have to learn like, what are the strengths? What are the weaknesses? They're essentially teaching you to become a peer reviewer for journals, right? You learn how to kind of distinguish things, even if it's not quite your line of research, even if it's not quite the study designs you do, you still want to have some knowledge of how to unpack these things. I will unpack a little bit of the article, but not a ton, honestly. I'll probably just do a glance through and like maybe just guide you through like what I see, but I'm going to do that later. The main thing I see, there are 4 main issues I want to talk about with how this article was reported. The first thing I want to talk about is that the findings were a bit sensationalized just from the article for the public essentially that talked about this. Sensationalize the findings a bit. So I want to talk about that. I want to talk about the issues with generalizing it to humans because that's part of the sensationalization is they didn't really, the title is more clickbaity. And if you're in the know and you click in and you find out this study was done in mice, you might go like, oh, it's a mouse study, of course. But that's not often how these things get reported. So I want to talk about that issue of generalizing mice research to humans because this is where the path of scientific misinformation, I think, starts, is sensationalizing and then people just assuming that something we saw in mice automatically will connect to humans. And that is absolutely not the case. So I want to talk about that. And then I want to talk about the broader issue of like why these things get so sensationalized when people do science reporting like this on this type of article. And ethical considerations, additional ethical considerations I have given the current political climate in the United States, particularly when it comes to rhetoric around autism. So, and also, quite frankly, the fact that we have, a leader of the CDC claiming we will have answers to things, which is not how research works either. So we already have someone who doesn't understand research nor science leading things and assuming, like, I think he thinks he knows, which is really scary. Dunning-Kruger effect. I don't know if you know that, but if you don't know what you don't know, you can be a very dangerous person put in a position of power. Anyway, let's go ahead and get into some of this. And then maybe as a bonus, maybe I'll make that a 5th, like, little chapter bonus of, like, when I glance through the article. Not someone who does this type of research at all. I've never been in a research lab that does any sort of mice studies, but just someone who took the classes and dusting off my memory of those classes because it was like nearly 10 years ago at this point. So let's go ahead and get into these issues. the first link I saw anyway to this article was to ScienceDaily.com. They did essentially kind of the public dissemination of the information, science reporting kind of thing, right? Now, science reporting is tricky. I'm not saying it's not tricky. It's really hard to translate something like, you know, theoretical science that might have application years down the road. It's very hard to translate that to the public in a way that's both understandable and interesting. Right? So the title that they put is Autism Symptoms Vanish in Mice After Stanford Brain Breakthrough. If I go to Google and I just Google search autism cure from Stanford, what you can see in the headline, like in the clicks, right? The links for you to click is, you know, you see, I mean, something comes up for autism's Stanford Autism Center, which makes sense because they do have that at their children's health hospital and such. But you see like epilepsy drug may help treat autism symptoms right here. Vasopressin treatment trial for children with autism. So that's different. That's a clinical trial. So that's not the one I'm talking about. Stanford scientists successfully reverse autism symptoms. That's one of the ones I'm talking about there. So this one, if I click over to it, it has Stanford scientists successfully reverse autism symptoms in mice. But the mice gets clipped off before when you're just scrolling through the links on Google, like your Google search, right? So it might make somebody want to click. But what I argue is, especially nowadays with how many people get bombarded with information and they might go search it, they might not click on the link even, right? They might just look at the headline and just remember that Stanford scientists successfully reverse autism symptoms. If that title is the only one that's seen, now you have a pathway to misinformation, because now you have people going onto Reddit and talking about autism symptoms were reversed. What the heck? What the heck? There's a way to reverse it. What the heck? This is one of those things where it's like, we just need to be able to discern a little bit more about this and to have a healthy level of, what is it called? Skepticism. Have a healthy level of skepticism around claims. If we see a headline that claims you can reverse autism symptoms in mice, making sure that we're not falling for it and thinking it's in humans, right? Because there's a real big difference between mice and humans. So what the study did is they essentially used a mouse model, so some sort of gene knockout type thing, or they impacted some pathway genetically to induce autism-like symptoms in mice. Okay, so something that has been accepted as these symptoms are sort of autism-like. Okay, and then they injected I guess it was an anti-epileptic, or they injected some sort of drug into the area that got affected by the knockout. So they already kind of knew what they were targeting, and then they saw these symptoms alleviate. In mice, okay? In mice. Why am I focusing on in mice so much? Because this is a real big issue with like neuroscience reporting, especially more like kind of this animal model neuroscience stuff. When it gets reported into like general population, people drop off the mice part a lot. I see this happen with people with PhDs in engineering who do this. They kind of fall for it a bit. So it's not like you're like not smart if you don't realize that, but only people in the field seem to understand. Like when I hear these articles from the PhD training I had, although I don't have a PhD, but from the 2 years that I did, it's like I get immediately like my little red flag goes up for, that sounds like a mouse thing, right? Like I'm up on enough of this research to know we are not anywhere near having like an injectable drug that can alleviate all autism symptoms in humans. We are nowhere near that. And no, taking away vaccines will not stop autism from existing. Anyway, that's a flat out falsehood, okay? And we have so much evidence that anything seen in terms of kid had vaccine and then autism symptoms showed up, it's correlation, it's not causation. Correlation does not equal causation. It's very important to always, always, always remember that. And quite frankly, even researchers fall for it sometimes. They will fall for correlation instead of causation a fair amount. So it's something really important to understand. When two things happen to occur at the same time, it doesn't mean one caused the other. But the biggest issue with this when it comes to mice, there's a few issues here. One issue is you have to remember whenever researchers, unless they're like actual in the field in nature studying like wild mice behavior, when you're talking about studies like this, you're talking about lab grown mice. Okay. These mice are not like field mice. Okay. Out on the edges of your town or something. They are not like the mice who show up in people's houses as pests. They're not like wild ones. All right. Lab mice have had a long time to be like, they have many generations of being produced for the purpose of studies. These are not mice who have wild instincts. They've never lived in the wild. If you release them in the wild, they will die very quickly. They will likely get scooped up by a hawk or something very quickly because they don't know. They've never been outside of a lab, which is very sad, yes, but this is the way animal studies go. I'm just talking about practicalities here. I'm not going to get into so much of the ethical weeds of animal studies versus non, because that's a whole separate deal. So here's the big thing to understand about rodent studies and generalizing to the humans, in my opinion, is the lab grown aspect and the fact that everybody's brains, all animal brains, have adapted to be the best essentially in the environment for which that brain is used in, okay? With the kind of body and the kind of environment, the brain has become somewhat optimized for survival and for just life, right? So field mice have they live in a what we would call enriched environment, in the sense of they have to forage for food, and they live communally, same with rats. We'll get into that if I ever decide. I have a thing on ABA I could talk about with operant conditioning and evidence there. Let me know if you want me to dive into that. I really could, but I'm pretty sure I'm going to attract a fair number of trolls. So I'm honestly a little scared to do it. I'm a little worried about doing something of unpacking like issues with considering operant conditioning to be relevant to today's research. Just want to it's not that it doesn't exist. It's just that it's not the whole story. So it's not a very complete, complex story. So I just kind of want to dive into that, but let me know in a comment below if you watch this, if you would watch that. Please let me know, because yeah, I think it would attract a lot of people who don't like it. So but mice, rodents, rats, they're all very communal in nature. These animals are highly communal. They work together a lot. Just, humans are pretty communal too. So they do have that relationship with us in that sense, that their survival depends on the colony, on the community working together, which kind of makes sense. A single little field mouse, you wouldn't expect it to really live very long all by itself, All by itself. Okay. Sometimes I just have to let them out to get them out of the cycle, because that song was starting to circle in my head, and it's like, if I don't make that reference, it's going to be stuck in there. It's kind of part of my unmasking, but it's also like, let it out so that I can move on from the cycle. Okay, so that's just for me. So yeah, mice in labs, however, often get raised effectively on their own, right? They get removed from their mouse parents, which there's reasons for that, and I'm not going into them, but they're unpleasant. If you like animals, don't read about the reasons why. They separate out baby mice from the mom. So, they get raised, even in labs where they do like enriched environments, which not all labs do, it's still not the same. It's still not equal to like being out in the wild. Okay. Enriched environment means they have like a wheel to walk on and like maybe a few little like games here and there, like little foraging games for their food or something, right? Because that's something their brain is really made to do. And we know that lab grown mice, when it comes to foraging and stuff, if they're not given that kind of enrichment, their brains do develop differently than like mice who have that kind of enrichment. Not only do we have an issue with like lab grown mice not necessarily having the brain complexity of, for example, a wild mouse, perhaps, when it comes to the enrichment and how their reward centers work and things like that. Like, it really impacts that kind of development, okay? But we also have an issue with the fact that humans are not rodents. We are part of the primate world. So there's a lot of issues with how mice things, especially when they're looking into cognitive things or symptoms of cognitive things, mental health things, these sort of things, when they're looking into it in mouse models, there are a lot of issues with translating what you find in a mouse model to humans, because the complexity ramps up a lot Just jumping from like a mouse to like a pig would be a ramp up in complexity, or a pig to a sheep, or, you know, definitely sheep to like a monkey. You would ramp up complexity each time you would jump some sort of animal model. Okay. So this is one of those issues with that. Okay. This is why like findings in mice, it's like maybe interesting to researchers and maybe useful to humans, but we're talking about usefulness in potentially like a decade from now, maybe, if it happened to pass through a lot of other tests and other research designs and other like probably other animal models and, you know, then clinical trials and that kind of stuff. Like you have to go a ways to get to where it maybe would apply to humans. Okay. And there's a really big issue when you're talking about these cognitive things, especially like autism symptoms and things like that, because human complexity is what leads to, like, if you read the DSM-5, you read the diagnostic criteria for autism, how are you going to know if a mouse meets those criteria? You know what I mean? Let's do this for funsies, okay? Let's do a little exercise here, okay? So if we're looking at the complexity of like rodent behavior to humans and we're trying to determine whether a rodent or a mouse has autism. For example, in the DSM-5, you have to have 3 listed deficits in social communication, okay? Difficulty with social emotional reciprocity, right? Nonverbal communication, possibly. Developing and maintaining relationships with other people could be difficult. How are we going to assess that in lab-grown mice that don't really have access to the same mouse community that like wild mice have? Right? Can't really assess that so much. And also even with wild mice, do we know how mice show emotional reciprocity? I don't think we do. Let me know if we do. Put in a comment below if you know a lot about mice. Feel free to info dump on that. I would love to know. Anyway, another thing, at least 2 of the following 4 restricted and repetitive behavior interests. So stereotype speech, repetitive motor movements. Now that's something we can see in mice. And that is one of the things I think they said they saw in these sort of mice, which is why it's autism-like things. But you can have repetitive motor movements outside of autism. That can exist in other conditions as well in humans, okay? You could possibly see if a mouse is using repeated sounds in terms of echolalia, but we don't know enough about, I don't believe we know enough about mouse verbal communication to be able to say, like, is it actually echolalic or is it like just, are they repeating it and does it actually have a meaning to another mouse? I don't know. So once again, people who know about mice, let me know if I'm wrong. We might understand if they're using more repetitive use of objects than we normally see. The rigidity, adherence to routines, ritualized patterns of behavior, we can probably see some of those ritualized or rigid adherence possibly in mice, maybe, depending if they've like been well trained for certain things or been observed for a long time before they did the gene knockout. So they know what their routine was and then they saw it change. Maybe they saw something like that. Now, they did also, I believe this is one of the big things they saw in terms of the sensory piece, this increased or decreased reactivity to sensory input or unusual interest in sensory aspects in the environment that we can see in mice. That is a thing that can be seen in mice, okay? Also, things that might exist in other conditions in humans. It might not be autism specific, but that is something they can also see. But you see what I'm getting at in terms of like the cognitive requirements, the complexity of human social interaction and communication and language use and all of this stuff. This is stuff we can't determine in a mouse model, okay? So that's what I mean by there being an issue with generalizing directly from mice behaviors. Even if they say autism-like behaviors in mice, they're not talking about a fair bulk of what makes up the autism diagnostic criteria in the DSM-5, right? Because this entire section here is all about social communication, right? Like there's no real true assessment for are the mice actually autistic mice? We don't really have that. And like, total side note, I actually saw this. This is a little bit, I feel like it's kind of a wild take, but people who feel like vaccines might give their dogs autism. And I'm like, okay, listen, dogs don't enter society and work jobs the way humans do. Even service dogs don't work jobs the way humans do, right? They get paid in like love and affection and food and pets, you know, and scritches and stuff. So like when you're talking about social mild disability, even if dogs have something that would be akin to autism relative in the dog world to like what human autism would be, It doesn't matter. Okay, it's not going to affect them the way it would affect a human in general. So, for God's sakes, keep your dogs vaccinated against rabies. Please, at minimum, Floridians, specifically talking to you, please, you do not want to mess with rabies. You do not want to mess with rabies. You do not want to mess with rabies. Get them vaccinated, please. Please. Okay? Because dogs get into stuff humans don't get into. That's why we vaccinate them for that. All right, off my little soapbox for that. Let's get back into this study business here. So now we already have this issue, right? We have this issue of we can't really generalize this information to humans right now. Truly, we can't. But like you saw when I scrolled through how that article got reported, at least the initial little link that you don't quite, like the words that if you don't click through, you might not see, this was in mice, right? So automatically we have an issue with, I think, general population doesn't quite understand the issues generalizing from mice to humans, and the fact that, like, in my brain as someone who works with humans, I look at those studies and go, oh, okay, I guess, they're not going to apply to me and my work for a very, 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 very long time. Like, and they might not ever apply. So, interesting, sure. Useful to human knowledge? Yeah. Useful for advancing certain things? Probably. Potentially useful in humans? Maybe. We'll see in 10 years. But like outside of that, if I'm not in that field as a researcher, I'm just like, okay, all right, maybe. It's just a big fat maybe, and I'm going to scroll on through. But I think when it comes to scientific literacy, general public, I wouldn't expect them to have that literacy. I wouldn't expect them to understand if they saw mice in the title that they think, oh, this isn't a cure for autism, right? In humans. We're nowhere near even beginning to think of that as this applying to that, really. Not really. Now, the other issue, other than the fact that I have a little cowlick that keeps showing up, let me brush that aside because that's annoying me. That aside. I mean, it's grammatically correct, but what a weird way to say it. Let's say you do understand that there's an issue with generalizing from mice to humans. Let's say you do. But if you scroll through, if all you're doing is looking at the little like words before you actually click to see the end of the title, for some of these articles, you might not see that it's about mice. You might just see the little preview and then think, oh, okay. And like, I think if you scroll through that enough, even very intelligent people, even people with some level of scientific literacy, might internalize the message of, oh, apparently Stanford invented a cure for autism, so I guess that's that, and it's like, no, they didn't. And now it just sounds like researchers don't know what they're talking about when they say like, or people with scientific literacy are not to be trusted, right? Which also leads to this general mistrust of researchers because there's not enough literacy in how research works and how academics work, and kind of the, quite frankly, to put it in, I guess, modern social media terms, clout chasing that researchers kind of have to do, quite frankly.
I said what I said.
They have to chase clout. That's what academia is. It's a clout chase in order to get grant money and things like that, right? So they got to kind of chase the clout a little bit and the attention. So then things get reported certain ways, things get spun certain ways. The actual researchers themselves might not have intended it to be that way, but it still happens. And then you end up in this situation where now it looks like the researchers lied. And it's like, in reality, it's not so much they lied. It's more like it's more like false advertising, right? It's like things get reported falsely enough, or things get clickbaited just enough because it's all about driving the traffic. It's all about the engagement, right? Especially for publications these days because there's so little of it. And ad revenue is mostly decreased. And this is why journalism is dying in this country. A whole other soapbox I could go into, but let's not. Let's move along. I've already started a little bit down this pipeline of why things get sensationalized, but let's go into that just a little bit more, okay? Now, this might be a little bit of a like academic bias on my part, but sometimes when I see these things coming out of places like Stanford or Harvard, these really big name universities, I think, ugh, yeah, here we go again. All right, yeah, these guys are always in the news, And a lot of that is because there's a lot of benefits for the universities, quite frankly. There's benefit to the university itself, putting out these articles and promoting these like results from these labs as being these big deals, right? It means something to Stanford if you just scroll through and the only little bit of the preview text you saw was scientists at Stanford found a cure for autism. If that's all you see, you're thinking, wow, Stanford, those guys are so smart. Stanford's so awesome. There's that aspect, right? You might not think that it really has any like application to the university, but it does. It's applicable in the sense that, first off, obviously people want to get into Stanford. We already know this because Stanford, Harvard, all these big names, like people want the prestige of going there. It means something to their degrees and their future careers. In certain fields, it can mean a lot, especially in sciences and research and like graduate school. It can mean a lot to somebody to get into like big name places like that. There's also the thing of like all universities, regardless of how big their funding is, regardless of how big of an endowment they have, they still want donors to their university, right? Anyone who went to any kind of college in the United States, I guarantee you there's probably at least one building on your campus that's probably named after somebody, right? And that somebody is either probably like a founder of the college or also more likely, I would say, a donor. Someone with a lot of money, maybe an alumni or whatever, and they graduated, they donated a ton of money, and now they have, you know, a whole department or a whole building named after them, okay? So yeah, money matters, especially in the United States, because we underfund everything anyway in education, and universities are not an exception to that. They tend to be underfunded enough for the kind of things they have to do, the kind of maintenance they have to do on their buildings, like just the upkeep, all of this stuff can be a real problem. So private schools, public universities, it doesn't matter. They all fundraise. They all want donors, okay? So getting their name out there in the news more often means more money pouring in. And it also helps to keep that cycle of prestige for these really high level institutions where they have alumni who went into really big fields, who made a whole lot of money, who can be really big donors. And then that their success means that they want their children to go there so the children can continue to have the success. And you have this whole like kind of Nepo baby kind of cycle that happens with that sort of stuff. Okay, that's just a thing that happens in those sort of fields and in these kind of universities. So it's essentially clout, right? It's the same thing as like social media clout in a way at like a more lower level, right? If somebody rage baits to get all the clicks and go viral and then become like a TikTok affiliate or something, and now they're gonna like always comment on things to get clout, right? There's also the benefit for the researchers in the sense that their name gets better known, right? Among other researchers. Name recognition is a big thing with researchers, okay? It's a big deal to be recognized by fellow researchers in your field, and it has something to do with also, in the United States anyway, how professors attain something called tenure, which means very, 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 very difficult to fire them. Nigh on impossible, possibly, okay? But in the United States, most of the time, if they get hired into any kind of professor position, they're called an assistant professor before they have tenure. In the United States, this isn't true in all countries, but in the United States, they're labeled as an assistant professor. Now, if they're in a field where it's hard to get a professorship, they're going to want tenure, okay? So if they're on tenure track for the first 5 years or so, can change the years depending on their name and what they did before they arrived and all this other stuff. But on average, about 5 years probably, they'll be on tenure track, meaning they're expected to produce a lot of publications to bring in a lot of grant money. And they're also expected to further their name because it furthers the clout of the university. It's all part of that cycle and connection. So that hopefully more money pours in, basically. It's kind of all about the dollar bills, y'all. You know what I'm saying? That's kind of how it goes. It's how a lot of this stuff goes. And so that is another thing. So name recognition becomes a big part of how they get tenure as well, right? So getting a lot of news and stuff drummed up about their results is a beneficial thing for the researchers as well. And even if they're tenured, it's still beneficial because they might get invited to talks and they might, they still have more, there's more ways essentially to show off in the academic world. There's more markers of like success and things like that they want. Okay. It's not just that. I mean, it can sometimes just be excitement over like, hey, we got results and it's awesome, because for some researchers that sometimes it's surprising that you got results and hey, awesome. Let's share it. Yay. But this is what I mean by like there's an incentive to sort of spin the results of the research into something that's clickable, into something that generates traffic for scientific publications, for that sort of stuff. This is what I mean by that, is that there's sort of this like little cycle that feeds into itself that I think most of people outside of academia, outside of the general populace, wouldn't really know about this cycle that happens, right? And it's good. I think that's honestly, I personally feel it's part of scientific literacy that's really needed a little more in our society, just in general, because understanding this cycle, that there is sort of this rhetorical cycle that happens and how things get kind of spun and sensationalized, it's a big part of that, which is really separate from like, for example, like political climate and what politicians might be trying to do or kind of policies they're trying to push. These two cycles are technically separate for each other, but they absolutely can feed into one another, right? Because if you have researchers or scientific reporting that says they've already cured it, and then people are like, I heard that they cured autism. Why isn't everybody cured? Where's the cure? Then it sounds like there's suddenly a conspiracy that there's no cure going out to people, and it must be all about, big pharma or whatever, which would be weird because I'm pretty sure pharmaceutical companies could make a lot of money selling that shot. So I don't think that's really how that would go. You know what I mean? It's like, that's where you get this sort of idea. And it's like, well, maybe you heard about a cure, but maybe it wasn't really a cure, right? Maybe it was just a sensationalized article from a mouse model that never generalized to humans. And so it just sort of fell off by the wayside. But if people remember, I saw somewhere that there was a cure. Why don't we all have the cure now? Someone's covering up that cure. Now it sort of generates that whole conspiracy theory kind of pipeline, right? Versus it was just sensationalized scientific reporting that inaccurately reported it and sort of sunk into people's memories as there's a cure now, when what they meant was there's a cure in mice, or maybe even lower level, there's a cure in a fly, in Drosophila flies. Right. And it's like, okay, well, that's not going to generalize to humans anytime particularly soon. Right. And then these cures never actually formulate. So then people think that something's being hidden when actually in reality, it's just poor scientific reporting, sensationalization, and then, maybe an accurate memory for things possibly among some people, or online discourse, people purporting certain things that aren't really real. And then the cure never manifests in 10 years. And it's because it just never generalized. So that's just something I definitely wanted to add into this, because that's an important part of scientific literacy when it comes to digesting this kind of messaging. That gets me to more of the additional ethical concern that I have with current political climate in the United States when you get something sensationalized like cure for autism found at Stanford, or whatever. If that's all you see when you're scrolling through things, if you're scrolling through Reddit and you only see that little headline and that's all that sticks in your brain, it can sort of seem like misinformation to the public. It can seem like researchers can't be trusted, right? You can develop this general mistrust in scientists because the cure is not actually being produced. You can develop this pipeline of conspiracy theories, and then that can be hijacked for nefarious purposes by certain political parties who have their own interests and who go down this path toward this sort of genocidal rhetoric that we see. So I think currently, right now, I was more shocked by seeing this. If it was any other political climate right now, I'd be like, oh, this is another one of those articles that shows up every once in a while. But given the current political climate, I just don't think this is we need to tweak the sensationalization, basically. Don't do that clickbait. Any scientific reporter out there, if you have the power to do this, I'm sure it's the editors and the people in charge of the money that probably really have more of a say. But anyone out there who has any chance of do I change the wording of this article to maximize SEO? Click no, please. Do not do that. Just put in mouse model, or put in potential cure in 10 years. Put in something a little more true that, it might not get clicks, but at least it can't be hijacked by nefarious political parties that actually want to that actually like have a very strong eugenics vibe, maybe we could just not go that way. That'd be great. So I think I covered all the main things I wanted to talk about. Let's go over to the article, which I have not really looked at, but I did pull up. Let's go over here if you're still here. If not, you can skip ahead to my wrap up section. I'll definitely try to mark that in a chapter so that you can skip this if you want to. Like I said, I'm brushing off a 10-year-old knowledge of how to read these articles, so please don't come at me if you're a neuroscientist. It's been a while since I've looked at this. But here's the article that they're talking about in Neuroscience, okay? The title is Reticular Thalamic Hyperexcitability Drives Autism Spectrum Disorder Behaviors in the, I don't know how they actually say this acronym. It might be CATNAP too, I'm going to guess. It kind of looks like CUNT-NAP, like KNIPT-NAP model of autism, okay? So that CNT-NAP2 model, that is the genetic model they're using. That is the genetic pathway they're targeting to promote the autistic behaviors in the mice and then to cure it with the drug injection, okay? So this part right here, this is called the abstract right here. Oops, sorry. There we go. There it is. This is called the abstract, okay? This is the quick little summary. So here's their they give a little background. So every abstract has a little bit of a background. So they talk about it like this. This is still background here. When they say thalamocortical circuit dysfunction, I believe they're talking about the neurological pathways from the thalamus to other cortical areas in the brain. Okay, now the thalamus, I will try to put up a little picture for you, but it's essentially this little relay center that's it's kind of here-ish, but you have to go like way deep in. It's in the midbrain kind of area a bit. That is what they're talking about. Same thing with reticular thalamic nucleus. Nucleus means you have cell bodies there, so that's like neuron bodies. Okay, so that's what that is. All right, I know it sounds really fancy, but they're talking about the body of the neuron cells. So if you see like kind of your general model of a neuron, I'll try to put it up here. This is like little neuron 101. You have like the little body guy, and then you have the axon. Okay, so nucleus or nuclei are essentially little collections of the bodies, and then their axons will be going out. So they're calling this, the reticular thalamic nucleus, a key modulator in thalamocortical activity. Meaning probably either ramps up or ramps down, okay? It's like a light dimmer, okay? It's either increasing or decreasing the activity of these connections from the thalamus to the cortex, okay? That's what they're talking about there, all right? And this is what they're doing. They're examining how this particular modulator, okay, this particular light dimmer, all right, handles or how it relates to these autism-related behavioral deficits by using they're going to knock out this particular gene expression in a mouse model. That's what they're doing. So this is all essentially background, and it gives you a little bit of like why they're doing this study. Okay, so this section of the abstract is background. All right, so this is how they say, this is what they showed as the autistic behaviors. Seizure, susceptibility, locomotor activity, so like motor, their muscle function is a little different, and repetitive behaviors. Okay, so as you can see, like I said when we were looking at the DSM-V criteria, these mice are only meeting the criteria that we can effectively just observe in the mice. This doesn't necessarily mean the mice actually are autistic mice, okay? Autism is a human diagnosis. Not for dogs, not for mice, not for any other animal, it's human. So what they did, electrophysiological recordings, my guess is they probably implanted little electrodes in their brain in those little neuron areas so they can record how those neurons fire, because neurons use electricity. Well, there's two different ways neurons communicate with each other. They communicate with electricity being generated by it's chemistry, essentially, that generates the electricity, the electrical charge, and also chemical. Chemical is from neuron to other neuron when it comes to like the neuron that's sending a message, then sends out certain chemicals to the other neuron to help that neuron develop its electrical charge, essentially, to make it sort of ramp up to send out the message some more, okay? So the chemicals are called neurotransmitters, neurochemical transmitters. Those are the little guys. That's the stuff like dopamine, okay? When you hear about like dopamine, serotonin, like that stuff, those are neurochemical transmitters between neurons, okay? And then within that neuron, it can ramp up its little electrical charge and send off that message, and it just repeats as the message goes down along your other neurons, okay? And what they're measuring is that burst. They're measuring the electrical burst. When they have electrodes put in, they're not measuring the neurochemical transmitters, they're measuring the electrical burst. That's what they're talking about there, okay? And accompanied by elevated T-type calcium currents. Okay. This is calcium currents. You're getting into what's making the electrical charge and kind of the nature of the electricity. So you have ones that either like burst firing is like, it's like a repetitive burst, you know, it's kind of like a neutron star when they see it pulsating. Okay. So you have that sort of thing. Depending on what's driving the electrical current, you can have these different ways of it developing the thing. So anyway, this is getting into the neuroscience weeds. You don't need to know all of that stuff. All right. Now, this is where they're getting into their results, okay? These are the fancy in vivo fiber photometry, I'm guessing photometry, confirmed behavior-associated increases in RT population activity, notably pharmacological and chemogenic suppression of RT excitability via da-da-da-da-da. So they're using a calcium channel blocker. I think this is the drug that got reported as the epilepsy medication. The pharmacological is a medicine, okay? Chemogenic suppression, meaning, okay, the medication is going to block this channel blocker. It's going to block the calcium channels that are creating that burst firing, it sounds like, okay? So it's blocking the ability for that message to be transmitted to the neuron that's going to have that burst activity. It's just, it's turning it off, okay? It's like really, really turning down the dimmer on the activity here, okay? Through medication, all right? Okay, and then they said that, okay, when they inhibited this particular one, this guy, if they activated the inhibitory, which is a dimmer, the light's going down, it's turning it down, right? So the more you turn up the turn down, does that make sense at all? That was a weird way to say it. But anyway, you know what I mean? The more you darken it, the more you turn down the lights, all right? That's what the activation of inhibitory. Inhibitory is turning it down, okay? Significantly improved the behaviors that they saw in the mice, okay? All right, so then they're finding that maybe, just maybe, this could be a potential medical target in terms of medication, okay? They're looking for a pharmaceutical target. They're not even talking about a cure, right? They're just talking about a target of these particular behaviors that they saw in mice, which is fair. That is good scientific reporting, but this is also a peer-reviewed article, and this is the part of the peer review process where if the scientists are trying to go a little too far with their claims, proper peer review would mean the peer reviewers would say, hey, you can't claim this is going to cure anything in humans, number one. You can't claim this is a cure for these behaviors either, because all you're doing is delivering a medication that might turn down the behaviors, right? So that's kind of what they're talking about here. And so this is why you don't see anything about a cure. You see them as highlighted as a potential therapeutic target. Potentially, maybe, possibly 10 to 15 years down the road, this would be proven to be a good target for a medication for humans. That's pretty much what they're talking about. Okay. So now that I've sort of parsed through the abstract, quite frankly, I'm just going to skim the rest of this jazz because, you know what? They were going to go into the neuroscience weeds, and this is not my area, and I don't know all of this stuff, so I'm not going to bother. But what you see, these things look really fancy, these figures, right? I remember looking at these being like, oh my God, this is what I don't understand when I first started looking at these sort of things. Okay. But what they're showing you, this is how they're recording the neurons. This is the thing they've implanted in the mice so that they can record the electrical current. And this is the output that they get, the like raw output from these recordings, from these little electrode recordings. Okay. They're showing you seizure stages here. This is probably more neuron activity, probably showing you like seizure versus non or something to that effect. Myoclonic jerk, that is like a big muscle reaction, essentially. These are all statistical things, seizure scores. These are statistical plots. Okay, so this is the math. They've done the stats, all right, on this sort of stuff. Usually what you see in these sort of articles, the very first figure is essentially them proving that their setup works. The way they're collecting their data is working. That's usually what they're showing you here. That's why they're showing you stats. These aren't results of the actual study. This is just, hey, and they did these behavioral tests. They did the grooming, reciprocal interaction. So these are the behavioral tests they did for the mice. The 3-chamber test, open field test, 3-chamber, grooming, and reciprocal interaction are the 4 things they probably tested in behaviors for the mice. Okay. So that's how they were assessing if the behaviors had changed. Okay. So this is literally them just proving like, yeah, what we did worked. That's what they're saying. Our method worked, because that's the first step in a lot of these articles. You got to prove your method's working. Okay. Now you get into the hyperexcitable circuit after the knockout. So they're showing their knockout worked. Okay. The genetic pathway, they successfully did what they were supposed to do. Okay. This is showing the burst firing in the neurons. So this is a horizontal slice, I'm assuming, of tissue there. But yeah, these are all neuronal firing burst patterns and the analysis they've done on those. Okay. And then you've got inhibitory output is altered. Okay. So now you've got the whole, this is probably getting into more of their results now. Okay. But similar thing. You're seeing this is electron output. This is the electrode measuring the electrical output. This is statistics. This is probably demonstrating the location, I'm assuming, of the injectable, because I think they injected the medicine. And so that's showing that, or no, maybe it's showing that, oh, this is the photo. This is a photo image thing. Okay. Anyway, that's showing you that stuff. Yeah. So it's all just them proving like, hey, yeah, we did what we said we did, and we got the results we said we got. That's pretty much all those fancy plots are showing you. Okay. It's a much faster, easier way to see results, especially for researchers who don't have a lot of time. This is a much easier way to digest the results with these sort of plots in view than it is this sort of text. Okay. Like, most researchers aren't going to dig into this text unless they're trying to replicate the study. Otherwise, it's like, I'm just going to look at the figures and see what they got and move along, because this is when they explained so in results in scientific journals, they have to explain all their results. So these are all the words essentially that explain what you're looking at. And it's a lot to read. Okay. If you think it looks like a lot and it's kind of thick and heavy and like dry, it is. It is exactly that. So actually, pro tip, a fair number of researchers skip that. They just look at the plots. They move right along. More plots, more plots. So in the discussion, this is where you get their summary of their findings and how this might apply to research, where they're at in their field, how this forwards the field, how it forwards our knowledge of things, right? That's all in the discussion. So this is their reselling of like, this is important stuff for us to know. We should publish this result, okay? Which is the thing, because you got to publish, right? So this is just basically confirming what they said in their abstract, that yes, they found that at least in these mice in this particular model, if you can turn down the reticular thalamic cortical connections and stuff, it tends to decrease the autistic-like behaviors in mice. So these repetitive behaviors they have and such, and the motor behaviors. Okay. And then they're breaking it down by all the little bits that they investigated. That's what they're doing here. Okay. That's that article. And then of course they always have to put in limitations of the study. Okay. So this is some of it's getting into the weeds. You know, they might say like, yeah, we didn't do this particular method. And yeah, we could have looked into that, but for reasons we didn't do it. And sometimes reasons, as fancy as they might put it here, some of the reasons might be like, we didn't have the money to do it, or we had mice that we were going to do that on, and then something happened, and like those mice were not feasible. Like we didn't have enough of them to like really do the test, or whatever it may be. Like, it can be many, many reasons. The main graduate student doing the study graduated, and we decided to go ahead and publish this now. Sometimes that's the reason, you know? Basically, the article itself is not sensationalized, okay? In conclusion, these researchers didn't mention, I think if I word search human, No, they didn't put the word human in their text. They have one citation that has human. They have some citations with it in the titles. It's not in the text of the article. They're not talking about humans with autism at all. They're not talking about autistics at all. They're talking about just mice. They're keeping it kind of in their lane. They're interpreting their results within the lane of this is basic research. It's nowhere near the level of applying it medically to humans. They didn't even talk about that. All right. So they did a good job. And some of these other ways these guys reported it, like I like this one, you know, a lot of them did a decent job explaining it, but this one, the main issue is that this in mice at the end doesn't show up unless you click on the article. So that's an issue, right? Because if you just see Stanford scientists successfully reverse autism symptoms, that's what you see in this title before you click on it. So if you're scrolling through and you don't click, you're not going to know this is in mice. So suddenly you think it's humans, and it's not humans. It's nowhere near humans yet. Okay. Same thing with, oh, there's a big ad thing. Same thing with this one. Stanford study shows autism-like behaviors can be switched off. If you don't see the in mice, you're thinking, oh, this must be a thing. Same thing with this one. This one doesn't even have mice in the thing. Experimental epilepsy drug shows promise for autism treatment. That doesn't even say in mice. You have to click and look at the actual, like, subtitle. And once again, I'm not blaming the authors for doing this because it really could be like editors and like other, you know, higher level people at this publication who like hold the purse strings. They might be the ones changing this. Okay. But this is a problem because we have this title, and if this title gets clicked around and shared around on social media and that's all people see, they're going to think, oh, cool, well, they have a treatment now, so great. We don't have to even have a debate anymore. Why are we even debating about whether autistics deserve to exist? We have a cure. And it's like, oh my gosh. It's almost like nobody ever watched any X-Men movie ever. Guys, X-Men handles the ethical implications of curing people, all right? And eugenics and such. So don't come at me if you don't understand what X-Men was trying to say in terms of that stuff. As far as I know, with the current state of science as it is, if you see any article or anything that purports to cure autism or to treat or to eliminate autism symptoms, it is not true. That is not where we're at with autism research, with the neuroscience, with the physiology. We are nowhere near understanding it to the level that we would need to be able to actually do that in humans. 9 times out of 10, what you're probably seeing is a title that is a sensationalized title reporting on a study done in like some type of rodent, probably mice, rats. Sometimes it can be flies, people. Seriously, sometimes it doesn't even have to be mammals necessarily. But like, that's probably what you're seeing. If you're seeing anything that looks fancy neuroscience-y, like fancy neurology stuff, and we have a cure, it's like, probably not. And also, I don't anticipate there ever really being one for autistics who are alive, because we cannot completely rewire a brain that grew its neurons in utero. When you come out, some of those neurons are just how it is, okay? Neuroplasticity can make subtle little local changes in a brain, but it can't just change the entire brain, okay? It's like improving traffic flow in a city, okay? Putting in some yield lanes, yield turns or some traffic circles or something. That's neuroplasticity, all right? It's not leveling the entire city and redesigning the whole thing from the ground up. That is not neuroplasticity. That's not how it works. And there's no medical anything that can do that. There's no drug, there's no surgery that can really do that for people. Despite what, some soap opera plots might have told you, yeah, we don't do brain transplants to change out the actors for for particular characters, okay? That's not a thing we do, all right? Okay, so hopefully that helps a little bit. Hopefully this isn't too long. Hopefully I edited this down a bit. But I just wanted to give a little more insight into how I digest these things when I see them, and what I see is the broader societal issues with how these things get reported, because it does really bug me. If you would like me to do more of the science-y diving into misreported science or what might be sensationalized science, drop a comment and let me know. And if you have something you've seen then link it. You know, you can send me a link through messaging or whatever, or you can click over to my website or something and drop me in a contact form, just drop the link in there and be like, hey, here's some misreported autistic science. Like, or could you give your insight on this particular, what kind of science they're talking about here? Like, what field of science is this? What's the study design? What it should be? I would love to dive more into that sort of stuff for people, just to increase our scientific literacy around these things, because that is a very important part of avoiding eugenics-based rhetoric, is being able to digest what is sensationalized, and then when that sensationalization gets spun and gets treated and like kind of co-opted by a particular movement for nefarious purposes against a particular population of people. We got to unpack that sort of stuff, I think, publicly. So drop a comment if you'd like me to do more of that. Other than that, do your best to take care of yourself, do something fun, find yourself some joy, find yourself some hope in the world and in humanity and in the beauty of something, even if it's Tetris. I mean, Tetris Effect is pretty beautiful, you know, as someone who plays it a lot. All right. And in the effort of helping to create more connection and community, let's use our weird powers for good. So in that spirit, keep it weird, everybody, and I will see you next time. Bye!